Good morning, good afternoon, and good evening, everyone, and welcome to today's webinar titled Rethink Your Impurity Analysis Strategy. Dr. Thomas Kofod was scheduled to present today, but unfortunately he has COVID, so get well soon, Thomas. So we are instead joined by Ejvind Mørtz, co-founder and COO of ELISA. He'll be discussing the benefits and applications of mass spectrometry-based host cell protein analysis and how they can be applied in a typical workflow. I'd also like to extend a big thank you to ELISA for sponsoring this webinar and making today's event possible. I'm Andrew Davies from the events team here at scientist.com, and I'm happy to be your host today. It is now my pleasure to introduce our first speaker, Ejvind Mørtz. Ejvind, are you with us? There we are.
Yes.
Wonderful. And Ejvind, the floor is yours. Take it away whenever you're ready.
Thank you very much, Andrew, for the nice introduction, and welcome to you all. It's a nice surprise to be here. So my colleague Thomas got ill with the COVID-19, and I had to step in with short notice. So please forgive me if something is not clear in the presentation. I'll try to do my best, but I'll also be happy to take questions afterwards if you need that. I'll just go to share my presentation here. So here at ELISA, we are a contract research organization. We use mass spectrometry to help our clients bring in new biologics to the market. So we do a lot of characterization of biologics. We also do a lot of host cell protein analysis. So we think it is time to rethink your HCP analysis strategy, and this talk will go through why that is. We have analyzed many projects, and we often get the questions from clients if we could not use LCMS for release testing rather than ELISA. And these are typical in projects where there is no good ELISA available, or where you have a fast track development of your project. Could be a COVID-19 vaccine, for example. So my talk is divided into three different parts. The first part is why we should do HCP analysis and the importance of using LCMS. The second part is about the LCMS analysis and assay performance. And the third part is several case examples from application in CMC development of new biologics. So first, I want to share you this press release that we did last week. It's a collaboration between Bavarian Nordic and ELISA. Bavarian Nordic is developing a COVID-19 vaccine. It's a complex product. It's a virus-like particle-based vaccine. There was no HCP ELISA available, and with short development timelines, there were no time to develop a process-specific ELISA. And then within 6 months, we managed to validate the method and do release testing under GMP, and the project is now in clinical phase 3. So this was released without using any ELISA at all. So how could we do this? I'll share some more details on this. But first, I'll give a little bit more background. So as a CRO, we here at ELISA believe that you can make superior medicines by using better analytics. And the analytics that we have at hand here are mainly LCMS methods, and we have developed them and optimized them for different CMC applications. We have more than 20 years experience from helping a lot of different clients. We have run more than 300 HCP projects using our LCMS analysis. We have had more than 1,000 clients spread over Europe and North America. And now we have the GMP certified HCP analysis. So how should you test and approve your product? I think most of you know these ICH Q6B guidelines on the test procedures for new biological products. So they have been widely adapted by different regulatory agencies, EMA in Europe, FDA in the US, and many other regulatory bodies across the world. So these guidelines, they describe how you should characterize these impurities. So there are basically 3 types of impurities as listed here. So there are the process-related impurities, there are the product-related impurities, and then there are the product-related substances. So the process-related impurities can be further divided into different types. They are the cell-derived impurities. So these are host cell proteins, for example, and host cell DNA. There are the cell culture derived. So these are things from your growth medium, inducers, antibiotics, media components. And then there are the downstream process derived. So these could be different processing reagents and leachables from columns, for example. Then you have the product-related impurities. These are defined as molecular variants of the drug substance without properties comparable to the product. So these could be aggregates, modified or truncated protein forms, precursors, or degraded products. And then you had the third categories. They are not regarded as impurities. So these are variants of the protein drug substance with properties and activities comparable to the product. So these are not considered impurities. They should still be measured, but they are not considered impurities. I will not cover these in this talk. So when we look for proteins, so here at AlphaLife we are experts in proteins. So these process-related impurities, where do we find the proteins? So these are typically host cell proteins or HCPs. They could be growth factors added to the growth medium to induce growth of the cells. These could be different media components, for example, fetal calf serum or human serum albumin added to help growing the cells. In the downstream process, proteins could be different enzymes added to modify the product. On columns, it could be affinity columns using different antibody affinities, and these antibodies may leak into the product. So these should all be measured. So why is it at all we should measure these impurities? There are 3 major reasons that you should know and remember. So the first and the most important one is the patient safety and the risk assessment of your product before you put it into the patients. So in the past 5-10 years, there has been a number of different examples where impurities have been immunogenic or caused adjuvant effects. So you raise antibodies in the patients against the drug substance. You don't want this unless you have a vaccine. Some impurities themselves might have biological activity in the patients. The second reason is for concern of your product. So you want to maintain product stability and efficacy. Some of these impurities might be enzymes that can degrade your product or modify it somehow so it loses its activity. Some enzymes might degrade excipients in the formulated drug product. Examples of these are different lipases and esterases that can degrade, for example, tween 20 that is used in many formulation buffers of many MAPs. And the third reason is for CMC purposes, for manufacturing consistency and product purity. So the CMC manager has to demonstrate the purification process clearance of impurities and document this clearance. You have to document the purity of your drug substance, and you have to document comparability between batches when you scale up, when you manufacture new batches, when you move the process to a different CMO, etc., etc. So all these 3 reasons are important. They are difficult to do when you only do an ELISA where you get only a number. So to do this in a detailed way, mass spectrometry has showed very efficient, and I'll show you some data on this. If we go and look at what has actually happened in the past 5 or 10 years, there was a paper summarizing different clinical events and experience with host cell impurities, where these impurities caused clinical delays or even complete hold of the product because of these effects I just mentioned. It's an excellent paper, and if you haven't read it, you should go and read it, because it's really changed the how this industry thinks about host cell impurities. There was also a second paper going through all commercial map products on the market, analyzing these by LCMS, and making long list of host cell protein impurities in these products. And so you can actually go and look up what has patient already seen in map products and how pure are they. There was also a third paper published by the Bioforum group in the UK by a consortium of many different biotech and pharma companies, where they have combined all the knowledge on different potential problematic HCPs in different products. In addition to the publication, they also have an online database where you can look up specific proteins and see if they are indicated as potential problematic, and what properties they are suspected to have. So this database is being continuously updated. So, and more knowledge is collected about these proteins. The identity of HCPs in your product, you can only get this by using of mass spectrometry. You cannot get it by ELISA. So all this information and these publications and these elucidations has only been possible due to the use of mass spectrometry. So due to all of this, the US Pharmacopoeia has a very good chapter on different analytical methods. The first chapter here on residual host cell protein measurement in biopharmaceuticals was published in 2016. It describes use of immunoassays, ELISA, HCP methods, reagents, and then it has some discussion and mentioning of orthogonal methods, so LCMS here, how that can be used to support ELISA development and documentation. This chapter has already been partly outdated, and that's why the US Pharmacopoeia about 2 years ago decided to write a new chapter. The working title is Identification and Quantification of HCP Impurities in Biological Products using Mass Spectrometry. So only mass spectrometry, no other ELISA methods in this chapter. So this chapter is written by industry experts. It will contain general guidance on what method and best practices to use. It will be a broad paper describing different possible approaches and recommended standards to use. And this chapter is expected to be released for public review early next year. So this chapter will be a guidance to the industry on how and when to apply mass spectrometry for impurity analysis. So when we look at a product going through the clinical phases, there are different HCP analysis strategies. So I've just written some of the major ones here. So if you look back, when you had ELISA only, this was the typical workflow. You would use a commercial ELISA kit if you could buy it for the early clinical development. Once you had got proof of concept for your efficacy, when you had locked your purification process, you would spend 2 years to develop a process-specific ELISA and then use that for release testing in your commercial product. This approach has the problem if the commercial ELISA does not detect all the HCPs, for example, an abundant HCP, and you find out late in the clinical phases, then you would have to go back and redo purification development and more clinical trials. So a high-risk approach only doing this. So, and that's why many companies starting using mass spectrometry as an orthogonal method to the ELISA approach. Mass spectrometry you can apply early on. You can use it to see if your ELISA detect all major HCPs. You can see if there are abundant HCPs. You can use it to guide your purification process development. And once you have completed this, you may go and develop your process-specific ELISA. In some cases, the commercial ELISA has actually been documented to be sufficient for also the commercial product if you use it alongside mass spectrometry. So this approach is being used now for most of the maps released on the market and many recombinant proteins. The third possibility is to use mass spectrometry only, as I just mentioned. This will be relevant for products where it's not possible to buy a commercial ELISA or where it's not possible to develop a process-specific ELISA. This can be because the product is too complex with proteins from many different sources. These could be cell and gene therapy products. It can also be products where the timelines are very, very fast, like the COVID-19 vaccines, or products where you have a fast track option with FDA or EMA. So this seems so obvious. So why has this not been done before? So why is it so challenging to do LC-MS analysis of HCPs, and why is it difficult to do in a quality controlled environment under GMP? So here I look at some of the different variabilities that you can have in a result. So first you have the sample variability. So this could be different matrices of different process purification samples. Sample complexity, you have other things in there that might disturb the analysis. You have the individual drug molecule. You have different concentrations. Then you have the sample preparation variability. We cannot analyze the proteins directly. We have to prepare the samples for the LC-MS analysis. This sample preparation has many, many steps. We need to reduce and alkylate cysteine, digest it into peptides, clean up the peptides, and this is all variable. The third variability comes from the LC-MS analysis. So this could be choice of column performance, injection volume, column temperature, mobile phases, and then you have the MS performance. This could be peak picking, MS signal integration, protein identification criteria, protein quantification algorithm, and this also alludes up to the data analysis variability that it is very complex. And on top of this, if you do this manually, you have the individual variability from manual handling of the samples and manual data analysis and evaluation. So you had to solve all these things before you get a reproducible quantitative assay. And this is what we have done here at Alpha. So I'll just go through the principle of the LCMS method we have set up here at AlphaLyse and applied for many projects. The principle consists of 3 steps, you can say. The first part is sample preparation to digest the protein into peptides. The second step is the LCMS analysis, and the third step is the data analysis part. So the whole sample preparation we have set up in a pipetting robot from Eppendorf. It has about 97 different steps over 6 hours. So it has digestion, denaturation, reduction and alkylation of cysteines, and all these complex steps to take into account this variability. We add intact proteins and we use these as internal calibration standards, both as QC standards and for calibrating the amount. These proteins we add into each sample before we start any sample treatment. So they will take into account any variability in the sample preparation step. The second step is a microflow LCMS system. It gives robust and fast chromatography. And then we have the data independent acquisition on this high-end mass spectrometer that can both identify and quantify proteins in low amounts, even though we have high amounts of the drug substance. We have a technical webinar explaining about this in case you are interested in more details. The third step is the data analysis. So to identify these proteins, we set up a process or product-specific database containing all the protein sequences of proteins that potentially could end up in your product. So these will be the host cell proteins, it will be the drug substance sequences, it will be any enzymes you add during the processing or other proteins that the sample may see in the manufacturing process. And then we use these internal standards for reproducible quantification and for quality control. It's explained on the next slide here. So what we see here on this slide is the 7 protein standards that we add into each sample, and we look at a dilution series of these proteins. So you can see on the graph you have the concentration on the x-axis and the sum of all peptides MS signal on the y-axis. So for each protein we summarize the MS signals for all peptides belonging to this protein. And what you can see on the graph is that there is a very good correlation between increasing amount and the sum of all MS signals. So for each protein standard, there's a very good linearity with a high R squared value. What you can also see that this line is different for each of the 7 protein. So each protein has its different response curve, and this is because some proteins they digest well, the peptides purify well, and they fly well in the mass spectrometer. They give a high response, and some give a lower response. Since we don't know what is the response curve for the individual HCPs, we use the median response curve of the standards to quantify the HCPs. So it's a median response calibration, you can say. So this means that most of the HCPs, they will be within the 2-fold range of this quantification. So when we give an amount based on the sum all MS signal, the actual amount could be half or double within this range. So that's for most of the proteins. Still, there will be some individual proteins that are even higher responders that will be outside the 2-fold range, or lower responders and outside the 2-fold range. So we use these standards to see if this is true for each sample and if there are any matrix effects and things not behaving as they should. And that's why we have 7 different standards to take this matrix effects and variability into account. The general method we have applied to more than 300 projects now. So a lot of different expression systems, mammalian, bacterial, yeast, insect, and plant cells, and also even some exotic ones not on this list. It has been used for recombinant proteins, maps, vaccines, and also more complex cell and gene therapies, or even bacteriophage products with lots of different proteins. So it's the same generic method that has been applied to all these different types of drugs. It has also been applied to different process samples, from the early harvest sample to the most purified drug substance. So if we look how this assay performs over time, so here we have looked at more than 150 different projects over 2 years, how the 7 standard performs. We look at the total sum all signal. You can see high variability. But when we normalize against the median response curve, you see that they all line up very nicely to the same line. So we remove a lot of this variability from sample to sample. There are still some outliers, and then you can go and investigate why this is an outlier. When we look at the individual protein standards and how they perform across the many different projects, you can see here how they perform. So this is from more than 1,000 samples. So when we look at the CVs of each of these standards, the inter-assay CV across different biologics and different samples, we have a CV of 11.8%. So highly reproducible. For the same sample analyzed again, we have the intra-assay CV of only 6.5%. So this becomes a very reproducible assay that has enabled us to take this into a GMP environment. So just to go through some case examples, how we have applied this technique. So in the early clinical phases, we often do these types of characterization assays analysis. So this could be documenting that the ELISA amount measured is actually okay. It can be more detailed characterization of ELISA reagents, standards, early process samples, what impurities are detected by the ELISA and which ones are not, and so-called HCP coverage analysis of the ELISA. We have quantification of individual HCPs, optimization of process development, batch comparability, IND documentation. For later clinical trials, we typically do scale-up comparisons and BLA documentation and PPQ run analysis. And then lately the release testing. So I'll just give you some examples of these in boldface here. So just to look at a typical purification process here, a protein produced in E. coli as inclusion bodies. So different purification steps. Here an enzyme is added to modify the protein sequence, and it's a 6-step process. So here on this slide, I have the HCP analysis results from the 6 different purification process steps. So you have the names of the protein, you have the theoretical mass and isoelectric point, and you have the amount in ppm for each of the proteins in each of the purification steps. If we look at one of the proteins, this small heat shock protein, you can see how the protein is cleared through the different purification steps. If this is not sufficient for you, you can use this as a tool to guide your purification process development, because you know the theoretical mass and PI, and your CMC experts can design and optimize a pH of buffers and columns and filters to target this protein specifically. In the bottom of the table, you see the summarized results. So on the lowest number is actually the HCP content in percentage here, or parts per million. So this is the number that's comparable to the number you get from the ELISA assay. So you can see in the step 6, the purified drug substance, you have a HCP content of about 400 parts per million, or 0.04%. So for process development, you can also use it for batch comparability. So here we are comparing the HCP profiles. 8 individual HCPs are compared across 3 different GMP batches. And you can see the green one, the most recent batch, is the most clean batch. An example from PPQ runs. So for a BLA filing, the client ran 3 different PPQ runs. Here we look at 2 individual HCPs, how they purify in the different steps. You can see the 3 runs are quite consistent, and you can also see the purification profile of the 2 HCPs are a little bit different, so depending on the protein properties. The next example is a method qualification for an IND application for clinical phase 1. So here there was no ELISA available, so we qualified the method according to IC8 guidelines, according to all the different parameters listed here. So so this is typically what you can do for early projects. So just to go through some overview thoughts. Why should you include MS-based HCP analysis, or should you? So what we can see from all authorities, publications, US Pharmacopoeia chapter about to be released, that HCP assays and use of them for development of biologics is about to change. It has already changed. There is an increasing focus on the use of LCMS for HCP analysis and for characterization of the ELISA that you use. There will be new industry standards on what LCMS workflows to use. That will be directed, and best practices will be described in the USP chapter. The LCMS results will enable you to do a risk assessment on your product if there are problematic HCPs in your product, and if they should be further removed, or if they are acceptable for your clinical trials. What we also hear from many clients that they have been asked by FDA and EMA to provide LCMS data to support the data they have on other impurity analysis. So regulatory authorities are already requesting this for some projects at least. And lastly, we have the very, very complex products where you have proteins from multiple sources, where it's not possible to use ELISA to measure all the different impurities. So all these problems we can solve, at least partly, by using of mass spectrometry. So here I have showed for you that we have one generic method that works for proteins from multiple different species and multiple different purification steps and buffers. The method can identify and quantify each residual protein and also the total amount. Even viral proteins can be identified and quantified. We have to set up a project-specific method containing the proteins specifically that could be present in your product and from your expression system. This method can be established within a few weeks. The assay is very, very reproducible across all of these different types of samples and projects, and it is available for validation under GMP if needed. So with that, I'll be happy to take any questions now that you may have. You may also contact me later via email or via LinkedIn if that works for you. So with that, thank you very much for your attention.
All right, Ejvind, thanks very much for the presentation, and also thank you for stepping in, of course, unexpectedly. We do have a bit of time left for Q&A, so I'd like to remind everyone in the audience that you can still submit questions using the Q&A tab on the right-hand side of your screen. And with that, you ready to answer some questions, Ejvind?
Yes, I am. So hello again.
Wonderful.
So
let's start with one here. We currently use an HCP ELISA and will need to bridge to new reagents next year. Can LCMS be applied for this?
yes, the short answer is yes. So the long answer is to bridge to a new ELISA. You have to do a new so-called HCP coverage analysis. You have to compare the old ELISA with the new ELISA. What impurities are detected by the ELISA? Are there differences? What are the differences? Then we recommend that you use the LCMS analysis to get the names of these proteins that are covered and not covered. You also want to know what HCPs are present in your HCP standard used for calibration of your ELISA standard. Does that match your early process sample? You also want to know what are the individual impurities in your purified drug substance. Are these impurities detected by the ELISA assay? And mass spectrometry can help answering all those questions. So we see mass spectrometry not competing against ELISA, but actually used as a supporting tool to select the best ELISA reagents for your project and your impurities, and also documenting that the ELISA is fit for purpose for your clinical trials.
Okay, great answer. This next one, pardon me, I think is a good follow-up to that then. You described them as complementary. How do the MS-HCP levels compare to ELISA levels in your experience?
So if you have a very good ELISA, and your standard fits your process and it covers most of the impurities, the number you get from the ELISA match normally quite well with the number you get from mass spectrometry. You can say the ELISA method, it is based on antibodies that are raised in animals. Some of these HCPs that are not immunogenic in the animal, so these HCPs might not be detected or only detected poorly. Mass spectrometry is a more, you can say, generic method. It's cleaving proteins into peptides and the peptides hitting a detector. So it's more generic. It measures more of the proteins. And then when we use these standards to quantify the amounts, it is a fairly accurate amount within this twofold range.
Okay.
So a good ELISA and a good mass spec assay gives fairly similar results.
Okay, wonderful. What? Pardon me. What is the ppm range for the linearity curve?
So the detection limit of this method goes down to the low ppm range. So typically we can identify and quantify down to between 1 and 10 parts per million for purified MAP products. Some of them are even sub ppm. We have a special sample preparation method for these, and we can go sub ppm. For other types of products, like vaccines, they are more dirty or crude. You have lots of HCPs, large amount of HCPs. Some of the COVID-19 vaccines, you have equal amount of HCPs as drug substance. So the upper range goes up to hundreds of thousands of ppm, depending on the product.
Okay.
So it has a linear range of about 4 orders of magnitude.
Okay, wonderful. I think that addresses one of our other questions too about the limits of quantification. A question here is, are there any recommendations for software to do the data analyses? And are there any publications about, I guess, data independent acquisition, I believe?
So software for this, different instrument vendors, they try to sell their own software. They also have independent software providers that just develop software. They all work in different ways. So for protein identification across all these different software packages, it is recommended that at least peptides should be identified in a protein in order to consider it as a positive identification. if you use at least 2 peptides, these different platforms will give you the same identifications. Regarding the quantification, it is more individual how people are doing it. We like here at AlphaLice to use these intact protein standards because they take in all this variability into account. Some other people, they might use synthetic peptides that are isotope labeled. They don't take the sample preparation into account. Other people, they use drug substance peptides that are also internally as calibrants. Yeah, so there is no, you can hear, there are different possibilities. There is no plug and play solutions yet that you can just buy and use.
Okay, great. Yeah, thank you very much for that. Next question here. How do you assess accuracy using the median standard curve approach when it encompasses 2 orders of magnitude?
So for these, here I showed on this, we evaluate the accuracy on these 7 standards. So we use the median response curve, and then we look for the deviation from the standard curve of the standards. How close are they to the median response curve? So you can give an accuracy for each of the standards compared to the median response curve. And when we make a dilution series, we can do that in the different concentrations. You typically do a high, medium, low evaluation.
Okay, wonderful.
If you want a higher accuracy than this twofold median consideration, then if it's one specific protein that's of concern, we can do more accurate quantification. This requires you have this as a purified protein standard. Then we can make the exact calibration curve and response curve for this protein. This could be an enzyme that you add during your manufacturing process. It could be, for example, benzonase, if you use that in your process. So there are different ways to make it more accurate. So here I showed results from the generic method that's immediately applicable. Jette, er du online? Er Andrew? Er vi på? I don't know if we lost Andrew.
Oh, there we are. Can you hear me now, Ejvind?
Yes.
Okay, perfect. Sorry.
I lost you somehow, or
Yes.
was I offline? I don't know.
Okay, sorry about that. Minor technical glitch. I think you already answered this question, but the next question, which this leads into, is it also possible to quantify enzymes added during process development? And then I guess the answer is yes.
Yes, it is. So, and we can even that often you have it recombinantly in high amounts, so we can do very accurate quantification of this one. And this is actually one of the typical problems that you may have, that if you add enzymes to your product, you also have to document the clearance of this specific protein to the authorities. So you have to have a specific assay for this protein that you are adding.
Okay, wonderful. I think you've touched on this question a bit already as well, but did you create in-house DDA, DIA libraries for all of the expression systems?
So the way we create these libraries, yes, we do. We do create libraries. So the way we do this is we run each sample. We create a library on each sample, not to miss the low abundant HCPs, for example, in the purified drug substance. And then we use this library to run the swath analysis against this library. So this enables us to do this very reproducible quantification of low amount ATPs. So we don't have a generic library that we use for all samples from different projects. We create the library on each different project.
Okay, Wonderful. Here's a great question. Are there any difficult sample matrices for the LCMS setup?
Yes. So, and that's why we have optimized this SAM preparation for many years to make it as robust as possible. Sometimes we still see these matrix effects. So, for example, if you have a sample containing glycerol, you may have some proteins that are adhering to the glycerol. You also have samples where you have denaturing reagents, for example, inclusion bodies, if you use guanidine hydrochloride to dissolve the samples. So these denaturants will also interfere with the analysis. So it's detergents can also interfere with analysis. So, and that's why we evaluate the performance of the standards on each individual sample to see if there are any matrix effects that we should consider. Should we optimize the sample preparation a little bit, or should we be cautious about accuracy that we report for the assay?
Okay, wonderful. Here's a great question. Is it faster to develop and validate an LCMS assay under GMP as compared to a process-specific ELISA?
Yes, it is. So if you have to go and develop a process-specific ELISA, that will typically take you 1.5 years, and then you have to validate it. So that's a couple of years we're talking about. So for mass spectrometry, the example I showed you with the COVID-19 vaccine for Bavarian Nordic, it took about 4 to 6 months for us to validate the method and do the GMP release testing. So that's definitely faster. I would say if you have an ELISA available and that's sufficient, it can be faster or just as fast to validate the ELISA assay. Mass spectrometry is still complicated. Yeah.
Okay, wonderful. Another question here. How do you manage identified HCPs that are below the limit of quantification value?
Yeah. So I think that's a great question. So we still, if it's identified with 2 peptides, we will still report it. We will also report the quantified amount, but we will say that it is below a limit of quantification. It is still relevant to have the information to compare batches. It can still could be a problematic HCP. In many mAb products, you have these lipases that could degrade between 20, even below 1 ppm. So you still want to know if it's there, but just below the 10 ppm limit.
Okay, we got, I think, another one here. What were the reasons for selecting intact proteins for calibration standards?
Mm. That was to take the sample preparation into account. So when adding these proteins before any treatment, they will take any differences in enzymatic cleavage activity, peptide purification efficiency. They will take that into account and the variability into account. And that's what also you see on these 2 plots, that when we use the standards to normalize anything, we normalize most variability away. There's no other way to get this kind of normalization.
Okay. I think maybe in the interest of time, we'll make, and getting everyone onto their next appointment on time, I think maybe we'll make that our last question. But I really wanted to thank you, Ejvind, for your time and wonderful presentation today and stepping in unexpectedly.
Thank you very much for helping this, Andrew, and thank you all for listening in today.
Okay, wonderful. It's been an absolute pleasure having you with us today. So finally, I'd also like to obviously extend a big thank you to our audience. Certainly appreciate your participation today. And finally, in closing, we really hope that you've enjoyed the Scientist.com webinar sponsored by AlphaLyse, and we hope to see you with us again the next time. Thank you, everyone.